我们第二位演讲嘉宾呢是陈薇老师，她是一位从学术界走向产业界的这个杰出女性代表，也是计算机视觉领域的知名专家。这个陈薇老师毕业于清华，然后博士呢是毕业于卡内基梅隆大学，期间呢也领导建立了该校的计算科技中心，之后又去了纽约州立大学奥伯尼分校任教。直到2018年，加入微软。他不仅拥有美国专利商标局授予的30多项专利，而且成果呢也广泛应用在 Bing、OpenAI 和这种 GitHub Copilot 等产品中。所以在这个信息技术中，真正是做了很大的贡献。那么他今天讲的这个主题是 AI for Microscope Image Analysis 3.0，让我们掌声有请陈明老师。
谢谢徐老师，也谢谢大会组织者，尤其是很多幕后的工作者，很多细节周到的帮助。因为我今天讲的东西呢，有很多算法，有些词汇呢，好像也只在那个英文的文章里看过，所以呢，我中文、英文相切不是很规范，我就用英文讲了哈，也比较规范一点。So the reason I want to talk about microscopy image analysis and the so-called 3.0, I also want to show that, as you heard from a lot of these talks and conversations, on the one hand, we are all making effort to advance AI. Meanwhile, I think all of us are also trying to adapt to the advances in AI so that we stay at the cutting edge and also our work stay relevant, right? Because the field is advancing so quickly and at a scale that's quite unprecedented with a previous technology. So what's the big deal about microscopy image analysis? Why do I call attention to this? So let me show an example. Hope you play. So when I first saw this data, I was fascinated. That was years ago, and some of you are probably familiar, have seen something similar data. These are mouse muscle stem cells, live stem cells in a petri dish. It's supposed to repeat, but it stopped. So it's moving around, so it's in vitro, and some of them are dividing, some are dying, some are differentiating. And when stem cells differentiate, that's actually the power of stem cell, right? That's why we do stem cell analysis, is that so that they can do tissue engineering to repair tissues that are diseased or injured. And since these kind of experiments can go on for days, maybe more than a week, and this is actually a sequence of images taken every 5 minutes in this case, and for some other cells, it's taken every minute. So you can imagine if that goes on for days or more than a week, there's a lot of data. It's impossible for a human to maybe go back. Maybe if I went back, then you can come back. Okay, it will play again. It's impossible to a human to really keep track of each individual cell, which is actually necessary for stem cell engineering, manufacturing analysis. And so currently what the scientists do is that they take a look at the local region and count the number of cells. They can't really track them. So what they need is actually what you see in this other one. You want to kind of identify each individual cell and keep track of them. The different colors denote different identities, and the little kind of tail are the recent trajectory of the cell movement. We actually want to keep track of that from the beginning to the end of the experiment, to construct the cell lineage. So here we can't show all of that because then it's really difficult to see. So we only show the recent history. And you can imagine how challenging this task is, right? So I was fascinated by this kind of challenge and also the very meaningful work, right, for stem cell therapy, and started working on this. And this is, kind of, see, the videos are always a challenge. Let's see if it plays here. Yeah, this is a zoom into a local area, right? This is in plane view of different cell identity, how they move around. That's, the vertical axis is time axis, so you see how the trajectory is being tracked. And, later on, you also see how when, the trajectory branch, that's a cell division, right, to analyze that. So in order to, bring more attention to this field, me and my collaborators started in 2016. We proposed a workshop to CVPR, the conference, And we were very grateful. At the first time, it got accepted as a half-day workshop. And there was a lot of exciting talks, a lot of interest with great attendance. And ever since that, we have been able to get this workshop accepted and organized at CVPR for 8 years already, till last June we had the last one this year. And through this process, we also observed a lot of development, and also kind of the field is also kind of shifting its focus because of the the advances in AI, and everyone is trying to kind of stay on top of the most exciting topic and also the latest approach. So I did something really trivial, just to show that this was taken all the talk titles from MIT speakers and accepted papers back in 2016. So see that segmentation was all the rage, right? People are excited about the problem, and there is learning. But even the keynote at that workshop was on model-based approach. That's even till 2016. And there's people are exploring learning and networks, and that definitely changed very quickly after that. This one was from 2022. So see that definitely deep learning, right? People are all employing that kind of approach. Segmentation is still a very important topic, and you see more and more attention to pathology. Digital pathology used to be a field that's really difficult to break into from technical perspective. Now there's more acceptance, more data available, and also more investigation. And another thing that really emerged over the past few years, until last year, is that multimodal omics, has been starting to get attention, with fusing different modalities, integration of that, and a lot of interest and investigation there. And as we all know, right, I think it's actually over the years accumulation, but last year somehow there seemed to be a lot of quite big breakthrough, right, in AI, both with SAM, like Professor Xu was mentioning, and also with language models. So then this year, when we had a workshop, there's further development. You see that the approach, instead of just mentioning learning deeply, even in myself, right, with my collaborators, at first we were even trying to innovate along network architecture, and by then we start to accept that let's just use foundation models. They are good enough, and focus our attention on really making a model work for specific applications, right? So that they actually can be getting closer to real-world application. So the self-supervised transfer learning, etc., was getting a lot of attention this year, and with the segmentation pathology, etc., still getting a lot of investigation. One other thing we notice is also this year, because there's also a lot of development in the microscopy technology. Image resolution has been increasing, and also I don't pretend to understand details like droplet-based technology for doing experiments and acquiring data has also advanced a lot. So this year, several of the invited talks are on single-cell analysis. Apparently now you can actually really study each single cell to really get further, deeper understanding of life, right, of ourselves, and a lot of related technology, leveraging AI, has also been forming. So similar to this, with the same progression, I also noticed, myself and our collaborators, our research has also been kind of, adjusting to what is the latest and most exciting. So from cell tracking, I also start to, with my collaborators, start to pay more attention to pathology image analysis. And like we're showing here at first, we also try to attempt to do some network architecture innovation. Later on, our attention has been, more on the domain adaptation. Domain adaptation, I think, a lot of the audience are probably, have more expertise than I do, is that you have a source domain, and with that, you have a sufficient, hopefully, training data to do, really good gradient-based learning. Meanwhile, there's always going to be new domain coming up, right? There's a domain shift, and also with new classes emerging. And we don't always have the luxury to collect, additional sufficient training set, because for biomedical images, as we all know, right, to get high quality annotation needs some domain expertise. So it's even more expensive than other kind of data set curation. So here we have been studying this unsupervised domain adaptation for quite a few years. Essentially, the goal is to alleviate burden, right, of very expensive data annotation. And unsupervised meaning that in the target domain I may have very few quote unquote, support data, meaning very few data annotated with 5 or 10 examples, something like that. But assuming in the source domain, I have adequate annotation that I can use that, right, to adapt to the target domain. And here, the challenge is, as you can see in this example, is that there's domain shift. Typically, for the target domain, it will be of something different, right? It all looks like pathology image, but maybe of a different, organ. So there's domain shift. And then the label sets, there may be some classes in common, but there will also be, new classes that are not covered in the source domain. So how you adapt the learning from the source, right, to the target, that's the challenge here. We have been studying this for a few years, and we saw that challenges also mean opportunity, right? So one thing, as we study, we start to realize that definitely current gradient-based optimization typically, right, needs sufficient data to guide the update steps. And then if there's more data or big domain shift, then the update can be noisy or lead to overfit. But the opportunities that particularly looking at a lot of related recent work, we realized that we can actually instead of, because we studied different approaches, now we are thinking that the adaptation will be more about optimization process. Can we transfer the optimization trajectory from the source domain to the target domain? And also one thing that's giving us a lot of insight that we want to leverage here is that for the stochastic gradients, the optimization tends to be low rank, and we want to exploit that, right, to do this from source to target adaptation or distillation. So here is our idea, is to essentially exploit the low rank nature of the gradient space and distill the optimization trajectory from the source, right, the teacher to the target, the student, and in that way provide external guidance, right, for the optimization in the target domain. This work just got accepted at ICCV this year, so it will be published. It's already in our archive. Will be published next month, presented at ICCV. So it's the first time I'm talking about this, kind of a preview. This is the overall design, from the source domain and to the target domain. As you can see, there's something in common, and also, quite a domain shift and the new classes. And the distillation, I'll talk a little bit into details. Here, overall, we call that dual stream distillation. One part of the stream is this cross-domain, right, from source to target, and cross-class distillation, because there will be new, novel additional classes. And here the steps, right, sounds, a lot of terminology. Really, it's about, exploiting the low-rank nature of the stochastic, gradient descent, so that, we distill the learning from the source to the target. And anchor classes, meaning, right, what's in the source domain. Those are, classes with adequate train set. So to exploit the low rank nature, we do singular value decomposition to identify the principal eigenspace in the source domain, and then do this low rank matrix approximation to select the top significant vectors, and then construct the principal subspace so that we can project the gradients onto the subspace and then minimize the discrepancy between the projected gradients. So that's the gist of the approach. There's definitely more detail in the paper, and also with the source code, open source. And another part of the dual stream distillation is this so-called self-distillation. We actually debated about the name, whether to call it historical or temporal. This historical here means what happened in the past. And the reason is that even with the previous approach, if the target domain bears quite a bit of a similarity to the source domain, the approach generalizes well. If not, it still doesn't work all that well. So how to make this approach generalize better? We also learn from related work that typically there's some connection between a very flat minimum and the generalizability. So we learn from that and try to kind of enforce our gradient steps to calibrate that. to smooth this optimization. Hopefully, that will generalize better. And the steps here, that's why we call that historical self-distillation. We took the past gradients, every so often in that optimization process to identify, again, low-dimensional substrates, try to, I guess, low dimensional is something that, more stable and we can understand better. So we, try to exploit that property to get, the principal gradient distribution of the local minimum. And then we project the current gradients onto these, the principal subspace to hopefully, smooth out the sharp and the noisy signal. And then we treat this projected gradient as a residual. So when we do the optimization, we actually sum up the original and the so-called rectified gradients to do the, the subsequent optimization step. So this is the other half of the dual stream distillation. And then we'll take a look at the evaluation. We looked at quite a number of data sets. Part of that was because of the review process. Reviewers, kind of like the result, but they are always skeptical. So we did a lot of experiments to show that it definitely applies to a lot of the data sets we can find. So this problem is about cancer tissue phenotyping. So the source domain has 5 classes. The target domain has 6 classes, and there are only 2 classes in common. So there's 4 novel classes, and it's a different domain. And the overall the task is about classification. And here the 5-shot setting is assuming the target class have 5 support. Essentially 5 data samples are labeled, and the 10-shot is 10 data samples are labeled. Overall, quite small labeled data set. The rest of the data sets are unlabeled, and the data are unlabeled. So here the color, the numbers that are colored in brown are previous best results from different approaches, all related work. And the last row is the numbers in bold from our approach. It is a dual stream distillation. And you can see that overall, I think a lot of them is quite a significant lift, for 4 or 5%, on both mean accuracy and also the mean FM metric. The metrics with the asterisks, they represent the metric that's calculated only on the novel classes, the 4 classes. And the rest are average across all the classes, including the classes in common. So this is one data set. And then we also take a look at the optimization trajectory. Remember, we are trying, overall, what is really happening here is to use a source domain optimization trajectory to kind of regulate the target domain optimization trajectory, right? Tries to do the adaptation there. So here, what's in the green color shows a quote-unquote, baseline optimization trajectory without this distillation of from source to target. And you can see that, first of all, it's kind of trapped in a local minima, and also there's a lot of sharp kind of oscillation, right? Not a very elegant, smooth, right, optimization. And then the one in blue color, first of all, it did reach the right minima, the global, within this context, and also relatively fewer sharp oscillations. So it's a bit better performing without the distillation. This other one is showing the quote-unquote flatness, right? We were saying that for the the better generalization, the local minima should be the minima should be relatively flat. So here the vertical axis shows the accuracy gap, and the horizontal axis shows the parameter perturbation. So the good quality optimization approach is that the oscillation should be small, right? Otherwise, it's very sensitive to different parameters. And you can see that the blue color come from our approach, and in general, right, it's a gentler slope, less sharp oscillation, in the performance gap, with the parameter perturbation compared to the baseline. So relatively better behavior. And then we show more quantitative evaluation. Like what I said, whenever nowadays we develop something, people always say, well, you cherry pick, right? On some tasks, it seems to do well. What about other tasks? So this is a nuclei instance segmentation classification task, meaning that you want to kind of get a contour of the nuclei and also classify, right? Recognize what that is. And here again, we evaluated with 2 settings, 5 shot and 10 shot, 5 or 10 support samples in the target domain. And also one metric is F1, one other one is called panoptic quality. Basically, it's a combination between for segmentation metric, the intersection over union, and also classification metric, with false positive, false negative, etc. So here again, the numbers in brown indicate the best previous performance, and the numbers in bold on the last row, the numbers from our approach. Again, overall, it's quite a nice lift, compared to previous approaches that, overall the approach still can, apply. And we also, evaluated on this other data set, that's skin lesion. I forgot to mention in the previous one, I think there was 2 classes in the source domain, 8 classes in the target domain. So there's 6 new classes. In skin lesion, it's a different kind of task. The source domain are coarse classification, so 2 high-level classes. And target domain was, I think, 6 or 7, I forget, finer level classes. So that means, there's actually no overlap in the classes, completely novel task. And also, again, with the same kind of, notation that our approach, performs quite a lot better than, all the previous approaches. So it's quite well behaved in terms of the applicability of the approach. And then we also did this ablation, right? People say, you talk about, fancy, design dual stream. What does each one do? Do they really contribute anything to the overall task? So here at the top row shows the full framework, right? The result, and then we kind of remove each component to see, if it matters. And it does seem to matter, right, to different degree. It does depend a little bit on the task, particularly the skin lesion data set is there's a lot of class imbalance, so the behavior tends to be a bit different. But overall, you can still see that, with the removing each part of the overall architecture does, reduce the performance. So it seems like, each component is still necessary in the design. And then here, we got challenged by the reviewers. They say, you show on microscopy image, but when people, to make it worthy of publication, what if I'm interested in other kind of data? So here we evaluated that on 3 very different tasks. One is a radiology imaging for pneumonia screening. One is, the fundus image, which is essentially, the image of the back of the eye to diagnose diabetic retinopathy. And the third one is something totally different, not in biomedical imaging. It's the office home data set to evaluate, from the artist kind of domain, rendering domain, to real world data domain to see if the approach still works. So again, here we only did F1 metric given I think at the time the time pressure compared to other approaches and the baseline. The overall performance boost still applies, and also quite a big lift for the natural image office home data set. So overall, it does really well. And here just show some pretty pictures. Qualitative evaluation, because numbers, sometimes what do they really mean? Particularly if it's a 1 or 2% improvement, does that really mean anything? So here, just for some intuition, this is on the nuclei segmentation and the classification task, because there's more things to see than just classification. So on the left is the data set in the target domain. Here's a ground truth, right, of nuclei segmentation. Also, different colors denote different classes. And our approach is the third column, and then with other kind of SOTA approaches from recent years. And you can see overall the behavior, right, even on a qualitative, intuitive level, also, it's much nicer that overall the both segmentation and also classification, right, is much closer to the ground truth. It's the different classes in these data sets. So I think just to conclude, in the interest of time, is that from our experience for this supervised domain adaptation, instead of just looking at the labels and the data itself, we pay more attention to the optimization trajectory to more do adaptation directly on the optimization itself to distill from the source domain to the target domain with a dual stream approach, right? One is more the domain cross class. Another part is from the self distillation, but more looking at the flat local minimum approach, right? Using the path gradients to project to the low rank subspace. And the paper is already posted on arXiv, so if you're interested, and if you attend ICCV, please stop by our talk and our poster. And with that, I will conclude in case there are any questions. Thank you for your attention.
好，看大家有什么问题 ？OK， 呀、
Yeah. Let me go to the slide. Slide, I think number 17. Yeah, that's the same task. Yeah, I showed. Yeah, same task. This is more the quantitative evaluation, and then that's the qualitative one. Yeah, these are all the previous approaches that they also tested on this data set. It provides pretty big lift across all the metrics, both for the F1 is more the classification, and then the panoptic quality is about both segmentation and the classification.
Okay, I have a question. Yeah, so your method lifted the performance a lot. But if you look at the absolute numbers in terms of F1 score, ACC, etc., it seems it's still pretty far from 100%. So what are the challenges in terms of reaching to 100% accuracy for F1 score?
Yeah, it's very tough. So here, actually, I think it's still because of lack of real high quality annotated data. Okay. So here the first one, the sub only as a baseline, the first row, it's actually if you train just on the novel classes. There are very few label data, so the performance is really poor. And then the second one is trained that across the new and old classes. And you see it's sometimes better, sometimes not even much. It's a really tough task. And then overall, people try to adopt from the source to target. It does somewhat better, but still, yeah, I agree, it's still pretty low. Particularly when you also combine with segmentation, you will see that the F1 does a little bit better than the MPQ, the panoptic quality. With segmentation, there's also the overlap of the contours.
I see.
So that part still suffers. Yeah, definitely. So even in some, from my other part of work that's not related to biomedical imaging, still that high quality, even not high quantity, but high quality train set still makes a big difference.
Okay, thanks.
Basically, the best so far we can do with 5 or 10 examples. And definitely, I think with the latest development, with SAM and those approaches, maybe we can also think about a different way. In addition to domain adaptation, can we also augment data through the generative model? Because currently, what's really is trying to tackle is that it's really expensive and hard to get high quality human annotation because it needs expertise. But then now, can we actually generate data from a few human annotated data set to generate more artificial train sets?
Okay, all right,
thanks.
thanks.
We're already doing that with typical, definitely with text, and also with some kind of images. But then with biomedical, I think it's just a matter of time.
Okay. Any other question? Okay, one more question, yeah.
Yeah. I think the next slide we also showed it works for images and radiology or other images. Because fundus imaging is essentially just a visual image of the back of the eye. And like what you said, it's actually very common because the stochastic gradient descent, there's a paper cited, I can show you in our paper in the references. There's a paper actually analyzed that. Stochastic gradient descent, actually it's very low rank. That's why we can use SVD to just get the principal subspace.
好的。